microbiome composition in patients with healed versus non-healed venous ulcerations. Chronic wounds tend to affect people from all different backgrounds, mostly though elderly people with underlying diseases such as diabetes, vascular diseases, and other autoimmune diseases. Also, people with Crohn's and IBS tend to be affected by this. There are a lot of research I found that these chronic wounds are often reflected by the skin's microbiome, which may either increase the chances of a wound healing or decrease depending on the type of bacteria that is present. So my project, I will be investigating this by looking for patterns between wounds with certain microbiota and how they tend to heal over a span of time. Due to COVID, these samples were not collected exactly how I would have liked because a lot of people were lost to follow-up for a multitude of reasons. However, there were some statistically significant values that were found after all the extraction and everything. So the first step of this process was to gather the wound samples. For each patient, there were 14 patients in this study. A sample was taken from the non-injured leg or the dry leg, that was how it was labeled, and the wet leg, which is from the actual wound itself. So the point of this was to show any overlapping microbiota, which would tell me that this was part of the skin's natural microbiome, or any alternate bacteria found in just the wet wound, which would tell me it was found from the air or another external factor, whether that be improper debridement or non-sterile technique being used. So first, I had to collect these samples. Next, after these samples were collected, we had to extract the DNA from the samples. I did this using the Norwegian Biotech DNA isolation kit. This followed a series of washing and buffing and separating the DNA from the walls and the rest of the cell to eventually have the pure DNA at the end. I then measured this DNA on the nanodrop to find the proper concentration of the DNA particles. After doing this, since they were all different concentrations depending on if it was the wound or the dry leg or depending on patients, I had to standardize each sample. So if the sample had too low of a concentration, I would leave it in the hood overnight to dry and increase the concentration. And if it had too high of a concentration, I would make a dilution to, depending on the groupings, I would do either 8 nanograms per microliter or 5. After doing this, it was time to prep for the PCR. I would add hot master mix, forward and reverse primers to the samples to then put them in the PCR and run 16S PCR. 16S is a sort of marker on the chromosomes that tell the type of bacteria that it is. This marker is generally unaffected by evolution. It has stayed pretty much the same for a long time despite any antibiotic resistance or anything of these bacteria. So it's a good marker of telling which bacteria it is. So run this protocol. Then I would run a gel to make sure PCR worked, and then eventually hand the samples to someone who would put them in the sequencer, the Illumina MySeq sequencer, to compile all the data into one file, which would be digitalized, allowing for easier statistical analysis using Google Sheets or using a protocol. So after doing this, a statistics manager handled these statistics and created graphs denoting whether or not certain bacteria were statistically significant in healed or non-healed samples. This is the table that shows time point A with the wet wound. This is just genus, so there are no statistically significant values here because genus is really specific, and it's better to open the span up larger to include family because finding these values with such a small sample size and just genus is not likely. As you can see, it's the same here for the dry values. Next, when you look at family, however, with the dry samples, there is a statistically significant value in the healed patients right here, as you can see. This number shows that this type of bacteria is increased in people who heal, so this probably lowers inflammation or fights off the negative bacteria. Here, there are 2 positive values for statistically significant, and these are in unhealed patients. So these bacteria are likely malignant and causing problems in the healing process, whether that be causing infection or fighting off the good bacteria. Thank you for listening.